Transmission electron microscopy, or TEM, is a powerful tool in behavioral neuroscience capable of magnifying objects by up to 2 million times, for high-resolution imaging down to 0.1 nanometers, roughly 2,000 times more powerful than the typical light microscope. By firing a beam of electrons through a thin sample and detecting the pattern of transmission and scattering, a sample's cellular ultrastructure can be resolved. Here, we will demonstrate the processing and imaging of the corpus callosum in a mouse brain by electron microscopy. After obtaining our fixed sample, we begin by isolating our region of interest. Although processing of larger samples is possible, as we're showing here, a brain matrix or vibratome is recommended to facilitate the collection of smaller samples. This will minimize the friction between the sample and the ultramicrotome knife in the preceding steps. With our region of interest isolated, we will apply the heavy metal stain, osmium tetroxide, which will bind electron-dense atoms to lipids in membranous structures and vesicles. The block turns black and brittle as the osmium tetroxide is reduced as it penetrates the tissue. Urinal acetate is then used as a second stain to enhance contrast to osmium tetroxide. It additionally preferentially increases the heavy metal density of some subcellular structures, such as DNA. The tissue sample is then dehydrated using increasing concentrations of ethanol. The epoxy resin used for embedding is highly water insoluble, so all water must be drawn out of the tissue. Once dehydrated, the ethanol is replaced with propylene oxide as it more thoroughly evaporates and can be removed from the sample more completely during infiltration. The sample is then left to cure in a silicone mold with epoxy resin overnight at 60 degrees Celsius. This epoxy will provide the sample with sufficient structural integrity to withstand the pressures of trimming and sectioning. A high-speed milling system trims excess resin by passing a rapidly rotating milling cutter across the surface of the sample. This step will also serve to minimize the friction between the block and the knife edge when sectioning. Using a glass or diamond knife, we take semi-thin 500 nanometer sections from the surface of the embedded tissue with an ultramicrotome. Periodically, survey sections are mounted onto a glass slide and dyed with the cationic stain toluidine blue. This allows for visualization of proteins at a high pH, and observation with the light microscope allows us to confirm sectioning depth as we approach our area of interest. Once observed, the sample is retrimmed to isolate only a small region that can fit onto a copper grid measuring 3 millimeters across. The ultramicrotome is then used to collect ultra-thin sections 50 to 100 nanometers thick. This is 500 to 1,000 times thinner than a standard sheet of printer paper. These sections are heated with a filament and mounted onto special grids which allow for the transmission of electrons. When mounted, the sections are stained once again with the aforementioned urinal acetate and with lead citrate, which interacts with proteins and glycogens to further enhance contrast prior to imaging. The sample is now ready for TEM imaging. The stained grid is carefully deposited into the sample holder, which is inserted into the microscope. Images are then obtained at 1000 to 2 million x magnification for further analysis. As you can see in this 8000x image, axons, the myelin sheath, and subcellular components such as mitochondria and synapses are readily visible with TEM.